Hi, my name is Noelle Pearce, and I'm presenting my research for my honors thesis in biology, Potential Effects of Influx of Cooling Tower Water on Meredith College Lake Organisms, Examining Survivability of the Water Flea Daphnia magna. This research was done with my faculty mentor, Dr. Pickering, and our biostat support is Dr. Pearce. So the motivation for this project came when I learned of a proposal to recycle leftover cooling tower water from the tower behind BDH into the Meredith College Lake. And that brought up the question, how will this affect lake organisms that I wanted to investigate? Water consumption is rising as urbanization increases around the world, so it's important that we learn to recycle our resources to be more environmentally sustainable for the future. Meredith College uses approximately 6 million gallons of water in the campus cooling system, with about 1 million gallons left over after evaporation. The water is released when it reaches a conductivity of 1200 microsiemens per centimeter, and at the time of this research, the plan was to redirect this water to Meredith College Lake, which would save about $36,000 a year. The image on the right is provided by Phil Corris, the energy manager here at Meredith, showing the proposed route that the water would take to get from BDH down to the lake. My research questions for this project were, does water type have an effect on Daphnia magna survival, and what chemical factors affect survivorship the most? So I did a preliminary study in spring 2023 using both Daphnia magna and Cyclops, both of which are freshwater crustaceans. The organisms were placed in 4 different solutions: 100% cooling tower water, 100% lake water, a 50-50 mix of tower and lake water, and then spring water was our control, as recommended by scientific literature. The organisms were checked periodically over 5 days, and dead organisms were removed. Our results showed no overall significant difference in the survivability of Daphnia magna or Cyclops across all solutions, and we determined that the Daphnia magna were easier to work. Water chemistry testing was done with the BrewLab Basic Water Analysis Kit by Lamott, and it tests for these top 8 factors: chloride, sulfate, total alkalinity, total hardness, calcium hardness, magnesium hardness, bicarbonate, and sodium. pH was tested with Lavator pH strips. So the way that these tests work is that a sample of water is placed in a test tube, and then a tablet or a solution is added to that sample. Then another solution is added 1 drop at a time to the sample until there's a complete color change. Each drop corresponds to 10 parts per million, so once there is a complete color change from red to green as shown in this alkalinity test, then I calculate the total amount of drops times 10 parts per million, and that gives me the total concentration of that chemical for that water type. Organism testing was performed October 16th through the 23rd for 168 hours. There were 200 milliliter solutions with 4 replicates of each, again using the 100% tower water, 100% lake water, a 50-50 mix of the two, and spring water. The organisms were checked 3 times on October 16th through 18th. On October 19th, I was only able to come in twice a day, and then on the 20th and 23rd, I was only able to come in once a day. We used dissecting microscopes and this light in the lab with a magnifying glass on it to help us find all of the Daphnia, and dead organisms were removed with a pipette every day. We kept track of how many living and dead organisms were in each dish by writing it on the note cards that are on top of each of the dishes. So this is a graph showing our results, survivability over time for the adult and juvenile Daphnia magna. The lines are the mean survivorship for that water type, and then the colored areas are the 95% confidence intervals. And the more that the confidence intervals overlap, the less of a significant difference there is in survivorship. So as you can see, cooling tower water is the only one that does not overlap with the other 3 water types, indicating that it had a significant effect on survivorship, and that survivorship was the lowest in that solution. When we split up the results by adults and juveniles, you can see that juveniles drove the survivability of the overall experiment. Adults have a lot of overlap with those confidence intervals, so they did not contribute any significant difference to survivability, while juveniles have a much lower survivability rate in the 100% cooling tower water. This is a graph showing our odds ratios relative to lake water. So basically, the closer that the odds ratio is to 1, the less of a significant difference there is. So cooling tower water is the only one that does not cross this null value of 1, and it showed a decline in overall survivorship by 33%. This is a graph showing the water chemistry values of each solution type excluding pH because it is on such a different scale than these other factors. But 100% cooling tower water had the highest concentration of all of these chemicals, which we expected because the water had been evaporated off so much before we used it in our experiments. These are the regression coefficients of the water chemistry factors. And basically, the larger the regression coefficient, the more significant effect it had on survivorship. So pH and magnesium hardness had the highest regression coefficients, having the greatest effect on survivorship. So limitations and recommendations for future studies. So we used adult and juvenile Daphnia magna. The Daphnia came in on a Friday, so I kept them in my apartment over the weekend. It is recommended that Daphnia have 24 hours to rest before being used in any experiments, but keeping them for a full 3 days likely had an effect on their lifespan and how long they were able to live once they were in the experiment. So it was more unclear, did they die because they were nearing end of life or because of the test solution that they were in. Also, some of the adult Daphnia might have gotten crushed during transfer. I was using a pipette to move them from the containers they came in into the test solutions. And some of the larger adults got a little stuck and might have gotten crushed. So that also would affect how long they were able to live once they were in the actual experiment. And then we recommend that future studies use only cultured neonates, meaning that we breed the Daphnia and use those babies in our experiment. This is because Daphnia reproduce asexually, having both adults and juveniles in the test solutions. The adults would start reproducing, and then it's impossible to tell which juveniles you started out with and which ones are the new ones that were just born. So to cut out that extraneous factor, we would recommend only using cultured neonates. We did not take into account how each water chemistry factor affected survival. We did calculate those regression coefficients, but we did not do any in-depth research onto how each of those factors affects survivability and affects aquatic ecosystems, so that would be something to consider for future studies. Temperature was not taken into account. All of our experiments were done at room temperature around 22 degrees Celsius, and it's estimated that the water entering the lake will be 24 degrees Celsius, which is about 75 degrees Fahrenheit. So this presents a risk of heat shock to the organisms in the lake, especially if water enters during the cooler months. We did not set a specific time limit for the experiment. We basically just ran it until all of the Daphnia were dead, but in future experiments, we would follow the literature and conclude the experiment at no more than 96 hours, just to, again, have some more control over the experiment and its environment. So overall, cooling tower water had a negative effect on the survivability of Daphnia magna. That much is clear. The plan to redirect the cooling tower water is no longer in effect, however. Mr. Pearce told us that they are adding some new chemicals into the tower water, so they don't want to release those into the lake. So this plan is no longer happening. But this study provides the foundation for future research on interactions between water chemistry and macroinvertebrates. It is not yet known whether Daphnia magna can represent well the sensitivity of other aquatic organisms, particularly benthic macroinvertebrates, which are huge bioindicators of ecosystem health. So careful monitoring is needed to ensure the stability and health of lake ecosystems as we move forward. I'd like to thank Dr. Cuffney for her input and advice on the methodology for this research project and for suggesting the use of Daphnia magna. I would like to thank Phil Pearce for providing us with a water sample from the cooling tower and answering my questions about the cooling system and future plans to redirect the water. Thank you to our biostatistician, Dr. Pearce for running statistical analyses and providing graphs and figures. And thank you to Dr. Pickering for helping me with this project and being a great faculty advisor and thesis mentor. These are my sources. And thank you for watching.